Okay, hello. We're talking about lipid metabolism and Parkinson's. I'm Audrey.
And I'm Bridget.
Okay, let's first start with Parkinson's. So Parkinson's is a form of neurodegeneration. And what this means is that when dopaminergic neurons, which are brain cells that produce dopamine, start dying off, you get symptoms like dementia, like difficulty speaking or moving. And dementia in Parkinson's is a really significant problem in the US. There's around 1 million people who currently live with Parkinson's in the US. And the number of patients have doubled since 1978. What this means is that the incidence rate of Parkinson's is currently on the rise, with 1.2 million people expected to have Parkinson's by 2030. And the problem is we don't have a cure for Parkinson's. We have treatments such as L-DOPA, which can handle symptoms of Parkinson's. It can slow down the progression of the disease, but we don't have an actual cure. And along with a lot of other scientists, what we're doing is we're investigating some of the cellular mechanisms for Parkinson's. Okay, so first, some more background information on the molecular basis of Parkinson's. What Parkinson's is it's caused by these clumps of misfolded alpha-synuclein proteins. And when these proteins are misfolded and clumped together in structures called Lewy bodies, they become really toxic. The toxicity ends up killing the brain cell they're in. But there's another layer to that. It's not just the protein, it's when these Lewy bodies start interacting with monounsaturated fatty acids. Through this interaction, they become more toxic and therefore kill the cell. So this diagram shows like a typical Parkinson's model where you have the misfolded proteins, they clump together, they interact with monounsaturated fatty acids, and then they become toxic. What we're trying to do is we're trying to take out that middle step, the monounsaturated fatty acid step. And monounsaturated fatty acids are produced primarily by this other enzyme called SAD1. So if we're taking out the enzyme that creates monounsaturated fatty acids, decreasing the concentration of monounsaturated fatty acids in the brain, we are hoping to alleviate some of the toxicity caused by misfolded alpha-synuclein proteins. So this is just another diagram of what our project is about. As you can see, when the proteins interact with our monounsaturated fatty acids, they become more toxic. Our goal is all about inhibiting the production of monounsaturated fatty acids. The next logical question is, of course, how are we going to inhibit this production? Of course. And that is actually done through competitive inhibition. And if you guys have taken AP Bio or Bio Honors, you may have talked about this already. But how this works is we have our enzyme, or in our wonderful analogy, a turtle, and it eats jellyfish. And that's the normal way the world works. What we're doing is we're introducing a plastic bag into the situation, something that looks a lot like what the enzyme is supposed to consume, but it's not quite it. So when we introduce a plastic bag into this situation, it stops the turtle, or our enzyme, from processing what it's supposed to, which is the fatty acid. Instead, with this new inhibitor, we're stopping the turtle from eating more jellyfish. And that's what our competitor inhibitor is about.
Okay, so now that Audrey has given you guys a pretty good explanation as to some background information, let's talk about what our project did that was completely new. So essentially, we're trying to synthesize 7 new and novel analogs of SAD1 inhibitors. Or in this analogy, that would be these plastic bags that are like kind of wrapping around the turtles. And so what we did is we looked at one specific compound, and then we just changed a specific part of that structure and looked at how changing that structure, or the R group here, would affect its efficacy in terms of inhibiting SAD1. So as you can see here, these are our 7 novel analogs. And the only thing that's really changing is that part of the structure that's in the red circle. And so again, we're just trying to look at how changing that red circle is going to change the compound's binding efficacy. And so we split up our project into 3 main steps. So first, molecular docking. So essentially what this step is all about is we are looking at how the computer thinks our compounds are going to bind to SCD1 and then look at their binding efficacies. From there, we are also going to further validate our results by using chemistry to synthesize all 7 of our analogs and again, test them in cells. See how biologically they would work. So here is what an example of some of our molecular docking results look like. This is pretty complicated, but what we're looking at here is our best working compound, p-toluol, which is that green highlighted compound over there, B or C, and it is bound to our protein, SCD1, which is this yellow squiggly thing right here. And as you can see, molecular docking is really useful because it tells us where exactly it's being bound to and what conformation it is. And so here's a here's a chart of our results. So this looks pretty confusing, but it's really not, because these negative binding affinities here just tell you that the more negative it is, the better your compound binds to SD1. Or in other words, the better your analog is attaching to SD1 or enzyme, the better it is at inhibiting it. And so we see here that we looked at our control, which was MF438, and that is a commercially available SCD1 inhibitor. And as you might notice, all of our compounds performed better than our control, which is pretty impressive considering that we're looking at commercially available SCD1 inhibitors here. And that's pretty promising. So this graph right here kind of just shows you those same results except in a graphical format. The blue circled compound is our p-toluol analog, and that was our best working compound. And as you can see, that structure right there has a smaller R group. It's a little bit hard to tell, but if you look at the compound that's right next to it, 3,4-difluorobenzoyl analog, you'll notice that that compound has a bigger R group. So Audrey's going to tell you some more about why these changes in R groups led to their different binding efficacies.
Okay, cool. So if you would look at this figure on the right, you can see that there's a green analog and a pink analog, and they're nearly identical in the way they're binding to SCD1. Those are our 2 best working compounds. Using molecular docking, we're able to understand how our analogs are chemically interacting with the different amino acids in SCD1 and determine why it's so effective at binding. And as you can see, because of this nearly identical overlap, we found a conformation that works pretty well. You can contrast that to our worst working analog, which is the 3,4-difluorobenzoyl analog, which is this whitish gray color. You can kind of see that there's this side that's protruding out from the ideal conformation from the green and pink analog. Using conclusions like this, using conclusions we're drawing from molecular docking, we can further modify the analogs we're creating. We can take a look at the R groups, the smaller R groups that protrude less from SCD1, and build on top of those analogs.
All right, so now that we've kind of established our molecular docking results, what we did next was we stepped into chemistry, and we synthesized our compounds. So this looks pretty complicated, but what it's essentially telling you is how we synthesize our compounds. It's our chemical route. And that first reaction is an acylation reaction, and then the second reaction is a Bock removal reaction. Third one is double condensation, and then fourth one is just a regular EDC coupling. We did run into a little bit of trouble with that last step, number 4, right there, but we are currently troubleshooting, and we're planning on using different reagents. Okay, and on to future research.
So really quickly, what we're doing this year is we're synthesizing the remainder of our compounds, and then what we can do is we can test them in real cells. So we treat our cells with our analogs, and then what we do is we extract the lipids from our cells. And using a machine called an LCMS, we can quantify how much monounsaturated fatty acids are present in the cells. Ideally, we'd want to see a significant decrease. And using those results, we can kind of pair the results we've gotten from our chemistry and from our computer modeling with the results we're getting from our cellular tests.
And so to tie this all back into why our project is important, what we're essentially telling you is that through lipid metabolism, we're able to look at how we can get a new cure for Parkinson's and potentially like save millions of people.
Does anyone have any questions?
Is there a reason that the commercially available inhibitor right now is so low in terms of efficacy? Like, is there like dangers associated with like having like a really high efficacy, or is it just like nobody has tried it before?
The danger isn't associated with the efficacy. We've kind of seen issues with like the synthesis route. So if we were to mass produce it, it wouldn't be as effective. And of course, we're just trying to build on top of existing literature to make existing analogs more effective.
Adding on to that, by looking at how you're changing the structure, you're able to create a structure-activity relationship, which essentially just tells you like how you can further improve your analogs in the future. Like,
Hi, Bridget. Hi, Audrey. Is your eventual goal to test this in Parkinson's patients?
So if we were like PhD students with a lot of money, then yes, maybe. With our resources, that is definitely not a feasible thing. I don't think we could get FDA approval for that. But if we found promising results, we'd hope that other scientists would be able to take information from our structure-activity relationship about which R groups might be the most effective and use that to build on existing knowledge about SCD1 inhibition.
And how long have you guys been working on this?
we started this project last year in ASI, so I would say like around 1.5 years.
I was wondering what made you specifically target lipids, because the presence of the other proteins that can be toxic, isn't it possible that if your inhibitors were not to get to all the cells or they weren't to function properly in some cases, that the proteins could still cause the damage? Like, why did you target lipids as opposed to the proteins causing the toxicity?
Yeah, that's a great question. So right now, Parkinson's and like a lot of other neurodegenerative diseases, the field is kind of dominated by like gene therapies, for instance. But lipid metabolism is something that hasn't really been studied that much until recently, like maybe 2012 or so. And so what we're trying to do is look at a new way to target like the protein for Parkinson's. And then your question was, why SCD1? Or
Yeah, so, well, if you look at like current developments in Parkinson's treatments, treatments that target alpha-synuclein directly or something like that are like rather aggressive, given that a lot of these mechanisms that we have that we're trying to inhibit are also essential for function. So it's really about finding a balance in inhibiting something that we need, so we can't inhibit it all the way, but inhibiting it enough that we see significant improvements in symptoms.
What are some ways people get Parkinson's? Like, is it only just born with, or?
So that's actually one a big thing we don't know about Parkinson's. We do know that there are some genetic links. There may be some environmental factors, but scientists do not know one cause of Parkinson's.